Hello, everyone. Thanks for joining us for C&EN's second annual virtual symposium, Advances in Drug Discovery and Development. I'm Lisa Jarvis, Senior Correspondent for C&EN, and I'm delighted to introduce our next speaker, Derek Lowe. Derek has been working in the pharmaceutical industry since 1989 on drug discovery projects ranging from schizophrenia, Alzheimer's, diabetes, and osteoporosis. But you probably know him best from his blog, In the Pipeline, where he provides astute commentary about the science and business of developing drugs. Rather than present a formal talk, we'll run this session like a Q&A. Just as Derek's blog inspires insightful and civil discourse about the drug industry, we're hopeful that today's audience will be equally participatory. Some housekeeping. If you want to submit a question, just click the green Q&A button in the left-hand corner of your screen. And if you need any technical assistance during the presentation, please click on the support button located on the top right of your screen. And if you want to follow along on Twitter, the hashtag is CENSYM15. Please note that C&EN does not endorse any company, products, or services that may be mentioned in this presentation. Finally, this talk will be archived and available on demand at the conference website for 3 months after the live broadcast, so until December 16th. While you get all your questions into the queue, I'm going to kick it off with the perennial question for the chemists in the crowd. Derek, would you tell students interested in chemistry to go into drug discovery today? And if so, is there a skill set you suggest that they develop or a path to take that would help them be the most successful in today's drug discovery industry? I'll turn it over to Derek.
Okay, that's a perennial question, all right. I get that a lot. And I guess my answer is that the skill set that you need is to be ready to change your skill sets, because this is, for chemists, for biologists, for a lot of scientists, this is a lousy time to be an average scientist in a high-wage country like the US or Western Europe or Japan, for that matter. We get paid a lot of money, and there are a lot of people in less expensive parts of the world who are willing to do what we like to do. And companies, of course, are always looking to shave every expense they can as long as the work gets done. So you need to be able to bring something that is not so easy to outsource. And in many cases, what that means is you need to be able to do things that are new. Whatever the latest instruments are, the latest techniques, the latest ideas, you need to be keeping up with them. You need to be keeping up with the literature. You need to be keeping up with your field. Because the days where you could say, well, you know, you should pay me because I can run lots and lots of Suzuki couplings, or I can set up libraries. A lot of people can set up libraries. A lot of people can set up Suzuki couplings. So that's not going to do it anymore. You really have to keep up with things. Have an answer when if someone comes to your office door and says, hey, why do we pay you? Why are you here? Why do we keep you around? You need to have, I guess, sort of an elevator pitch answer, a quick answer. I do this. I do this thing, and it's important for this, and you don't have anyone else who does it, or you don't have anyone else who does it as well as I do.
Great. Well, we're going to jump from a really broad question to one that digs a little more into some hardcore chemistry. A question from the audience: what computational chemistry advances do you think can or will have the greatest impact on drug design over the next several years? Back over to Derek, please.
Okay. Yeah, computational chemistry. I'm sure I have some colleagues who are groaning at the thought of me giving another opinion about computational chemistry. But even though I've had my ups and downs with it over the years, there's no doubt that, you know, it's driven partly by Moore's law, which is still moving along pretty well, although you wonder about the next few years. It's being driven by advances in software and advances in understanding, which come a lot more slowly, but they're still there. So it's getting better all the time, whereas other branches of chemistry are improving, but maybe not quite at the same rate. Of course, computational chemistry has a pretty high mountain to climb too, especially considering what people have asked it to do over the years. But one of the things I'm interested in watching is the free energy perturbation methods that some companies are exploring. People have been talking about this stuff for years, but it's been computationally sort of prohibitive to do. And it's still a major burden. You have to have some pretty serious hardware to make a dent with it. But I think that area has a lot of potential. I just don't know yet how much that potential is going to be realized. I'd like to see some real world applications. The good thing is, if people can get this to work, we're going to see real world applications because the companies that can do it are not going to be shy about telling everyone that they can do this. So free energy methods, I guess, are the first thing that I think of. Beyond that, there are, of course, a lot of other things under the umbrella of computational chemistry. But I think what the first thing people think of with computational work is, can you predict which molecules I should make? Can you rank order this bunch of possible analogs? And that's where I think the free energy stuff really has a potential place to work.
We've got a lot of computational chemists, I think, watching right now because we're getting a lot of questions on that topic. Here's a more general question, about epigenetics as viable drug targets and your thoughts on whether those are going to succeed. I'm guessing someone's referring to the epigenetic drugs that are in development to treat some forms of cancer. You want to weigh in on that? Do you feel comfortable on that one? We can go over to Derek. Uh-oh. It seems like we've lost Derek for a second, so we will try to reconnect him, and hopefully he'll be back briefly because I see the questions lining up in the queue.
Okay. All right, I'm back. All right, epigenetics. That is a tough area because it reminds me a little bit of the nuclear receptor area as it was, say, back in the late 90s, early 2000s. Actually, the nuclear receptor area, as it arguably still is, this is an area where the biology is extremely difficult. It's very interesting, it's very complicated, and it clearly is going to control a lot of stuff, but that's the promise and that's the peril because it controls so many things that it can be hard to separate out the effects that you're seeing with any given, say, small molecule acting on an epigenetic target. If each one of these targets is controlling dozens or hundreds of downstream events, genetically, that's a pretty big knot to untangle. And I don't think we know yet enough to say. I worry that it's going to be one of these things that's going to take another 5 or 10 years to really undo all these strands and figure out what the best way to go is. But of course, people aren't waiting 5 or 10 years. They're charging ahead. There are a lot of companies charging into this. I wish them a lot of luck. I think that some of these things are going to work, but some of them, I think it may be one of these cases where you not only have to be good, you have to be lucky. I mean, look back at the nuclear receptors. The best nuclear receptor drugs were the PPAR gamma compounds that were already in development before anyone even knew what their target was. Most of the nuclear receptor work that came after that during that boom time in the early 2000s ended up not leading anywhere when it got to the clinic or even preclinical. So it's something that, I don't know, I hope for the best, but I'm kind of glad that I don't have a lot of money tied up in that myself.
Okay, well, interesting response. So we've gotten a few questions about compound libraries and whether you think that the quality of the compound libraries that are in existence, either in-house or commercial, are good enough to yield, good leads. Maybe let me try to tie two of these together because a related question was, are large companies running out of compounds to screen in their high throughput discovery? So maybe you can weigh in on where we're at with high throughput screening.
Okay. Sure. I don't think there's any danger of running out of compounds. Compound space is, of course, gigantic. It's ridiculously huge. And we've got new synthetic methods coming online all the time. So I don't think we're running out of compounds. Now, as far as how good these libraries are and whether they're good enough to come up with leads, that's a tricky one. Because if we had our current compound libraries and we're running things, 35, 40 years ago, these would be spectacular. The kinds of targets people were screening then, I'm sure that we would light up things all over the place. But targets, of course, are not getting any easier. Occasionally we'll come up with some pretty good-looking target through biology and genetics efforts. Of course, the hope back when we sequenced the genome was that there'd be dozens and hundreds of these out there, but that hasn't exactly turned out like that. But a lot of the targets that we're looking at are getting trickier. And the kinds of chemical matter that are going to work for them are probably getting trickier too. You can find all sorts of suppliers right now who will sell you protein-protein interaction optimized libraries. I have no idea what a library of compounds optimized for protein-protein interaction is going to look like, except they're probably all going to be big and ugly. And of course, as you get to big molecular weights, the potential universe of compounds gets bigger and bigger and bigger. So your chance of sampling that space gets smaller and smaller. So I think that the compound libraries we have are probably a lot better than they ever have been. People are paying a lot more attention over the years to QC, to compound diversity, toward culling things that are frequent hitters, toward pains type compounds, all sorts of false positives. We're getting better and better at weeding that stuff out. But at the same time, the targets we're expecting these compound libraries to deliver against are getting trickier with probably more unusual chemical matter. So it's an arms race. Right now, I think that we are better off than we have ever been, but the question of is that good enough is still an open one. Depends on what kind of target you're screening. Some of it, for instance, epigenetics, the last topic that came up, some epigenetics targets, if you screen against BRD4, you will probably find some chemical matter. You can do a fragment screen and get perfectly good fragments. However, there are a lot of those other epigenetic targets you can screen all day. And as far as I can see, you won't find very much at all. So the answer varies. I hate to cop out like that. I mean, someone asked J.P. Morgan one time what the stock market would do, and he said the market will fluctuate. Whether or not your compound library is good enough depends completely on what kind of targets you're screening against.
Okay, this is a broader question, but the nature of drug discovery is such that you spend a lot of time writing about the failures, just because we see more failures than successes. But I wondered if there's ever been an instance where you've doubted a drug discovery approach or technology or target and been surprised when it actually works. Or I guess if there's anything that you're seeing in the pipeline that you're really hopeful is going to work, that is some really cool technology.
Oh yeah, I've been surprised, definitely. I guess I'm enough of a pessimist where I figure that most things are not going to work. So when something really does work, I'm very pleasantly surprised. But I have been. I've had some things that didn't look like they were going to work out that really did fly right. And they flew right at several different points. Some of them, the SAR opened up and suddenly we found a whole new series of compounds that we didn't realize was even there. Sometimes we would be very worried as we went into, say, 2-week talks because we were convinced that X and Y and Z would probably happen, and it didn't. Of course, sometimes something else happened, but yeah, I have been surprised on the upside. But of course, if you're going to talk about drug discovery, if you're going to give a more or less honest picture of how it really works, you're going to end up talking about failures because, of course, the clinical the clinical failure rate is still pretty much 90%. So there's just a lot more things that don't work than do for now. That's our big problem. As far as technologies and things in the pipeline that I'm really curious to see how they're coming on, I can cop out and say the immuno-oncology stuff is spectacularly interesting. The big question is, how far can it be extended? To how many different fields of cancer can we get away with this? What are some things about the immune system we don't know yet that we're going to find out as we start doing this? So the immuno-oncology stuff is extremely interesting. A lot of the biological and bio-small molecule hybrid stuff I find extremely interesting as well. There's, of course, all the RNA stuff out there. I realized, and I lived through the antisense period in the early 90s where it looked like antisense was just going to come in and wipe everything out. That was going to be it. Every single target you could think of solved by antisense. And that didn't work out. And I don't think that everything's just going to get wiped out by siRNA. But between siRNA, between the messenger RNA stuff that Moderna and other people are working on, between the cell-based therapies like the immuno-oncology and the gene editing type technologies, there's a lot of really interesting stuff going on. Of course, you notice most of these things I'm talking about have a strong biological component. And that's what I think medicinal chemists, we have to shove ourselves into this. We have to find a role in assisting these things. Biomolecule, small molecule hybrids, modulating these processes with small molecules. For instance, right now people are testing the T cell chimeric antigen stuff along with PD-1 inhibitors. We need, as medicinal chemists, to find wherever we can to make a home, to come to an accord with all this new biology, because otherwise we risk becoming less relevant in some of these areas.
I like that phrase, coming to an accord with new biology. And you bring up immuno-oncology, and that ties in nicely with a question from one of our viewers, which is, thoughts on, I don't think this is necessarily what you're referencing, but thoughts on what this, how small molecules can play a role in immuno-oncology. And, maybe I'll just build on that question. How significant of a role do you think they could or might play?
Yeah, I can't give too many details on this because immuno-oncology is not my field per se, although I'm an extremely interested outside observer. But just the combination I mentioned before with the CART stuff and PD-1, that is one role, and I think that might be sort of a prototype of the kinds of interactions we can look for. We need to find whatever immunological levers and checkpoints we can that might be amenable to small molecule control. Now, there aren't a tremendous long list of them, but there are some, and I think that we should be pushing with maybe some rather out there screens to see if there are other things that we might not think are amenable to small molecules, but might be. There are huge numbers of interactions that we really either don't screen for or haven't had too much success screening for in the past. And I would adduce protein carbohydrate interactions, which of course are important for immune response, as one of those. That might be an area to start pushing into and see. This gets back to the chemical library question. It might be that the kinds of compounds that most people have in their chemical libraries would give low hit rates for something like that. Fine. Go outside the box a bit. I hate using that phrase, outside the box. I should be beaten on the head for using that. But try some different chemical matter, some more unusual looking chemical matter, and see if you can get things to happen in this area. This is another one where if you're going to screen in some of these things, the existing libraries may not be enough of a fit. But we're going to have to find these things. Otherwise, biology has been making some really tremendous strides recently. And one good thing is to sit back and applaud them, which I do. The bad thing would be to sit back and grumble about, well, these people are there, they're pushing us out of the way, we're going to be outmoded. Don't be outmoded. Get up and do something about it.
Okay, well, we've touched on immuno-oncology, which is obviously a really hot topic. And so with a lot of success that we've seen, I want to kind of turn the coin to Alzheimer's, where we've seen a lot of failure. And a kind of chicken or egg question. Wondered what you're going to see first, a good mechanistic understanding of a disease like Alzheimer's that then helps us design drugs, or will we finally get some drugs on the market that help us then understand what's actually happening with the disease? Can you give us some of your thoughts about where we are in treating a disease like Alzheimer's and maybe more broadly other neurodegenerative diseases, and whether you think there's any technologies that are emerging that could help us solve some of these really tough questions?
Chicken and egg with Alzheimer's, chicken and egg, that's a tough one because when you think about it, the Alzheimer's compounds that have gone into the clinic, the ones that are in the clinic now, and the ones that are headed there, are mechanism based. They are target based because people are looking at amyloid processing for the most part, or they're looking at just sheer muscarinic signaling in the earlier generation, or even if they're looking at tau or whatever, these are mostly target-based, mechanism-based things. So as those compounds are in the clinic, then we can start saying something about whether or not these particular targets are really relevant to Alzheimer's. For example, you look at the gamma secretase inhibitor that Lilly had in the clinic and failed terribly. It actually seemed to make Alzheimer's slightly worse. You have people arguing that this wasn't a good enough gamma secretase compound, this wasn't a fair test and all that, and you have to worry, though, because that's a little bit like the no true Scotchman fallacy. A real gamma secretase compound would have worked. I don't know. That may have been a real gamma secretase compound. We now have beta secretase compounds. We have the antibodies that Lilly are trying desperately to get to work, that Genentech and others have tried. The amyloid hypothesis is not yielding as readily to this stuff as you might hope. And as with the gamma secretase, you could say, well, that's because the amyloid hypothesis is right, but we just haven't hit it with the right agents or hit it hard enough with these agents. Or you could say that the amyloid hypothesis is maybe not all there is to it. Don't know which way I come down on that. I'm going to stay agnostic on it. I used to work in Alzheimer's a long time ago, and if you told me that it was 2015 and we'd still be arguing about whether or not amyloid is the proximate cause of the disease, I don't know what I would have done. Banged my head on the window and tried to jump out or something. I can't tell you. The lack of progress has been very, very hard on people. So we're going to get some answers with the things that are in the clinic now. But if you want to find a compound that would just go in and help people with Alzheimer's, I guess you would need a phenotypic screen. And I don't see how to do a phenotypic Alzheimer's screen because there's not a good model for Alzheimer's on any level, not on the cell level, not on the animal level. I mean, they're better than they used to be, but is there anything that people are confident enough of to say, yes, this is what's going to drive our great big phenotypic Alzheimer's program? Given the cost, given the time, the expense, the effort of doing Alzheimer's trials, I don't think so. I don't think anyone's got that amount of nerve right now. So we're going to see what these mechanism-based compounds in the clinic do. But beyond that, we're going to have to come up with more mechanism-based compounds, I guess, because I don't see any way to just come in with any sort of broad-based, we don't know how this works, but it works type compound.
We have a few questions from people who are wondering about making the transition from academia to industry. People who are not, a grad student, but who have been working in academia for a few years. I wondered if you had any advice on that, whether it's don't do it or some specific steps or skills that folks might be trying to take in order to to kind of make that transition?
Okay, that's a good question. The first thing I can recommend is that there's a book that they should read. It's one that I reviewed on the blog some months ago, and it was all about making that transition from academia. It was a very well-written thing. It had a lot of very sound advice in it. The first thing I would do is, and the authors are going to love me for this, now I just wish I had the title to hand, but the first thing I would do is go and read this. It was from an academic group that has actually done this transition, and it has a lot of very good lessons in it about what surprised them and what they didn't expect. And there's going to be a lot of things that surprise you and that you don't expect. Not all of the stuff in the book is favorable toward academic science. You have to be ready for that. So I think you have to be braced for a different world. Industrial research is both more freewheeling than a lot of academics think, and also a little more rigorous than a lot of academics think. We don't just all sit around all day trying to figure out what color the box is going to be and what the slogan is going to be that we print on the sides of the balloons. It's extremely difficult, and you need a lot of people that most academic researchers don't have access to. There's a lot of very specialized work, toxicology, formulations, pharmacokinetics, on and on and on, that you really don't find these folks in academia as much, partly because it's very hard to run a PhD program in, say, formulations. So these are specialists who do this all the time, and they're very good at what they do. But if you've never interacted with them, it's a different world entirely. So if you're going to get into industry from academia, you have some studying to do. But keep in mind, all of us who are in industry, we came up through academia. So we know a bit of what it's like back in academia. It's just that academics often don't know anything about what it's like in industry. Read the book, though. That'll start you off on the right path. And then have an open mind. Be prepared for things not to be like your preconceptions. And be prepared for it to be very hard, because it is.
Okay. A general question about your blog. You've been doing this for over 10 years now, and I wondered if you could talk about what your motivation is to keep doing it. It's a long time to be writing about the industry. What keeps you motivated? Also, I wondered if you could talk about how your relationship with your audience has changed over the years. I think when I started reading you, it was in the early days of your blog, and there's such a civil discourse that's going on, and as you know, journalists and investors alike mine the comments to learn little tidbits about what's going on inside companies. So I'm curious if you could talk about just that relationship with your audience and how you've maintained it and how it's changed over the years.
Sure. Well, as far as what motivates me, um, I don't know. I like writing about this stuff. We can't rule out vanity either, but I like writing about this stuff. I've always been a pretty quick writer, but that is of very little use in drug discovery. Being able to give a good talk is nice, and it doesn't hurt at all, but it's not as of much use in drug discovery either, because I can't go down and talk to the cells in the dish or the rats in the cages and exhort them to do what I want. My speech making qualities just are kind of barren in that area. So I've decided that I should use whatever abilities I have to write and communicate this stuff to do it this way. And I've gotten used to it. In the mornings, I just feel bad if I don't write something on the blog, because most mornings I do. So the good thing is that material is always coming. Very rarely do I ever sit down in front of the keyboard. I write this on the train ride into work. Very rarely do I ever sit down and think, oh gosh, what am I going to write now? I usually have several things queued up. My browser, my kids make fun of me, my browser always has about 20 tabs open. I have things in email, I have stuff written down on little notes and etc. So I have things queued up to write about most of the time. So I just enjoy it. It's, some people drink coffee first thing in the morning. I don't even drink coffee. I'm pretty weird. My wife can't figure out how I stay on two legs. Instead of doing that, I write about some scientific news. As far as the relation with the audience, I think it's been pretty similar once I started to have an audience. It took a while. I mean, if I go back to the early days, it took a while before people noticed that there was such a blog, and readership has just been climbing and climbing over the years. I have been really, really happy with the way the comments section works. I have all sorts of other people, journalists and other people who run blogs and internet sites ask me, how do you do that? And the answer is, I didn't do it. The people who are interested in this kind of thing came to the site, and this is the sort of atmosphere and the sort of community that generated, and I am just really, really happy about it. There's not a lot of craziness in the comments section, for the most part, a few, but there's not a lot of ad hominem, not a lot of yo mama, to use the vernacular form of ad hominem. There's just a very high signal to noise. If you're not reading the comments on the blog, you're missing a lot of the best stuff. I just hope nothing ever happens to mess it up. And it looks like the comments section is going along pretty well at the new site, so I'm just happy as can be about it. Oh, I wanted to mention one other thing. Readers have chimed in about the book that I was trying to remember. Here it is. It's called A Practical Guide to Drug Discovery in Academia, the SPARK approach. So Practical Guide to Drug Development in Academia. That's the one to look out for.
I wanted to ask about something that you blog about pretty regularly, or maybe it feels regular since you've been blogging for so many years and I've been reading for so many years, but it's about the relationship between industry and academia. You seem to have some skepticism about the various approaches that companies are taking to try to dig in deeper to academia and these kind of open source efforts. I wondered if you could talk a little bit about things that you think aren't working and whether you think there are any models that are working about how companies and academic scientists can work together, whether they exist or don't exist, or are there things that you think could work?
Okay. Okay, one thing that comes to mind is you occasionally see the idea advanced that industry has all these wonderful drug candidates that are just sitting on the shelf gathering dust because we're lazy or we're not too smart or the managers lost track of what projects they were doing after the 14th merger in a row. There are a few things like that, I'm sure. But for the most part, I don't think there are incredible vast treasure troves of neglected drugs sitting around, in our cabinets. So that's one thing that I think some people need to think about hard if that's going to be their model, is we're going to find all this stuff that industry has just totally forgotten about. There will be a few. I mean, Homer sometimes nods, but there's not like dozens and dozens and dozens of them just sitting around. I have been skeptical over the years of some of the academic approaches into drug discovery, but I have to say it's gotten a lot better. One reason for that might be because there are, well, a number of people from industry who have ended up in academia. Some of them voluntarily, and some of them did it by virtue of needing somewhere to go. But the end result has been that there are more people in the academic chemistry and biology departments than ever before who really do know and understand something about real-world drug discovery. And by real-world, I'm afraid I'm using it as a synonym for industrial. Because, as I was saying in the previous answer, there are just a lot of things that are part of the process that you hardly ever find outside industry. But there are a lot more people now who know about this and who've had some experience with it, and the connections between academia and industry are thus stronger than they ever were. Now, we still regard each other with some mutual incomprehension and some suspicion, At worst, the academics look at the industrial people and think, here are these folks who don't know how to do basic research anymore. All they do is yank ideas from other people and go off and make a mint on it. And of course, the industrial chemists look at the academic ones or the scientists in general and say, here are these folks in their ivory tower worrying about grant applications all the time, never realizing how much actual work it is between a neat idea and a drug and how many places it can go off the rails. So we get frustrated with each other, but I think the two are actually, while they're not, hand in glove, they're closer than they've ever been, at least during my career.
So we've had a few questions from viewers about layoffs and downsizing in pharma. I mean, obviously your blog is a go-to source for folks to give tips when a layoff is happening, and a lot of us who are journalists read about those layoffs on your blog first. But could you just give a little sense of what the temperature is like out there? I mean, have we reached some sort of homeostasis in terms of big pharma layoffs, or you know, give us a sense of what that's like, and maybe if you have a sense of what the job market is like for, say, a graduate student who's looking for their first job in the pharma industry.
Sure. Well, as for layoffs from big pharma, part of the part of the reason things don't seem quite as bad is, my golly, they've already laid off so many damn many people. I mean, there's just, how many more can you cut? But as we saw when GSK turned around and closed Research Triangle, and when Bristol Myers Squibb turned around and closed Wallingford, there are more places to cut. So you really can't rule this stuff out. Um, Both of those had been rumored for a long time. The people who were working at those sites were always a little bit on the nervous side, excuse me, on the nervous side, because they kept thinking, oh, we're not at the mothership, we're going to be peripheral. But it was still a shock when those things happened, and a lot of people are still losing their jobs. So no, you can't say that the big pharma layoffs are over. And there could always be another big merger. Someone could turn around and pull one of these things tomorrow. Pfizer was after AstraZeneca so heavily. They could turn around and decide they need to go after someone else. Probably not someone in the U.S. because they still need to solve their tax problems. But in general, these things are always out there. That's the unnerving part of being in a public business. If you're working for a public company, someone can turn around tomorrow and make a run at you and decide to acquire you. Maybe they'll be spending too much money. Maybe they'll be making a mistake. But people have spent too much money and people have made mistakes doing this. It could happen anytime. So I wish I could say that layoffs are a thing of the past, but no, they're not. As far as coming into the industry, what that means is, I think, and I've been saying this for some years now, I think that the balance of people who are working in the industry may well be shifting. We've got fewer people, a somewhat smaller percentage working for the really big companies, and a correspondingly larger percentage working for a longer list of smaller companies. That's not necessarily better or worse, it's just different. And it's just different if you're going into that job market too, because it means, for one thing, you're probably going to be hopping around from company to company, because one of the things about these smaller companies is they don't all make it. They're a little bit like projects inside a larger company. They come and they go. So you'll need to be ready to move, hopefully not move physically that often, which is the other factor. That also means that one thing to keep a lookout for is you may well end up moving to the Boston, Cambridge area or the San Francisco Bay Area, maybe San Diego, etc., because there are more companies there, of course. That means if something goes wrong, you can perhaps find another job without having to call the moving vans. Whereas if you're working for the only company that's doing immuno-oncology in southeastern North Dakota, if that goes down the tubes, you're probably going to be moving somewhere. So it's nerve-wracking like that. There isn't really much job security in this business anymore. I think there used to be. When I was at a previous company, I saw someone who was celebrating like their 30th year with the company, and I thought even then, I'm never going to see this again. I never have. I don't know if we ever will. If you know that coming in and you're not surprised by it or dismayed by it, but you've already come to terms with it, it's going to go a lot smoother for you.
Um, so we all will move to Boston soon. No. uh, I wanted to ask about, kind of chemistry breakthroughs because, one of the things that you do really well is write about some of these big academic breakthroughs and how they relate to, your job. Things like, inserting artificial base pairs into living cells or Marty Burke's, synthesis machine. And so, I just wondered if any of these recent developments stood out to you as a technology that's going to change the way you do your job or change the way in 5 years or 10 years medicinal chemists are going to do their job. Is there anything that you really have just said, wow, that's super cool and is going to change the way that we're doing chemistry in the future?
Yeah, well, the two that I can think of, you mentioned the synthesis machine. Boy, did the comments section erupt on the blog when I started talking about that. As it is currently constituted, that machine is not going to come in and change medicinal chemistry. And even in a much improved version, it's not going to come in and remake the landscape. But I think that something like it, more automated type chemistry, is inevitable. We've had sort of a romance for a long time in chemistry that everything is done by hand and there's no substitute for adding everything yourself. In some cases there's not, but there are a lot of very routine reactions out there. Suzuki couplings come to mind, like are used in Burke's machine. There are a lot of very routine reactions out there, and in general, I think that something like that that can be done by a machine should be done by a machine. And there are a lot of bread and butter organic reactions that I think are becoming more and more amenable to being done by machine. Now, I first saw a machine doing organic synthesis when I was in graduate school. I was at an ACS national meeting and saw one of the old Zymark robot arms going around, supposedly optimizing a reaction. It wasn't working that well, but even at the time, I panicked a bit. I was in grad school, so I thought, oh my God, someone's invented the mechanical grad student. I better get my PhD fast. But it is coming. And I think it's a good thing. Running Suzuki reactions all day long is not very exciting. We should be using our hands and using our brains for something else other than setting these things up over and over. So automated synthesis, mechanical assistance is one thing. As far as sheer chemistry, I mentioned this on the blog the other day. One of the things I like very much is all the photoredox chemistry that's coming out. Some of that lets you do things that you can do by other means, just by a different and maybe easier or cleaner way to do it. Some of the reactions, though, are things that are not all that easy to do. And I think this field is just getting started. The redox potentials involved in these photochemical catalysts are very large. There's a lot of energy there. And I think if you pick the right co-catalysts and get the catalytic cycle working in the right way, there's going to turn out to be a lot of very interesting transformations. It has the potential to be as big a change as palladium couplings were, Suzuki couplings, etc., were in the late 80s, early 90s. So I'm very much looking forward to seeing if that prediction comes out.
Uh, well, speaking of hot technology, I wanted to bring back this CRISPR notion. I mean, I think you brought up the antisense craze in the 80s and that everyone thought that was going to blow away drug discovery and be the answer to all our problems. And obviously it hasn't played out that way. Could you talk to me a little bit about CRISPR and how you think it fits into, the drug discovery paradigm? I'd also like for you to talk about chemistry in CRISPR. I mean, what roles do chemists play? I mean, is it up to chemists to solve some of the delivery problems that might happen with getting getting that delivered? Maybe you could just talk about that a bit.
Sure. Well, CRISPR delivery is a chemistry problem, or a formulations problem, or a formulations chemistry problem. So yeah, that is one place that chemistry can definitely play a role. I think that there are some therapeutic areas that have the potential to be remade by this kind of technology, and others not, because of course CRISPR in the end is a gene editing technology. There are diseases that are specifically strongly tied to the genome, and there are diseases that are not. You can look in, the metabolic basis of inherited disease database and find plenty of small orphan type diseases that we know exactly the mutation that's behind them. Things like this eventually are going to be where CRISPR gets applied. And I suspect that the various companies out there doing it are looking to pick them off one by one. I'm not going to talk about somatic cell and germline type editing, because that's coming eventually. But just for now, the other thing though is that even with some of these diseases, it may be that the cell types that you need to edit are not going to be so easy to access, or to access cleanly, just because of physical problems, because of isolation from other cells, maybe because of cell turnover. Not all of these things are going to be as easy to do. The other thing though about CRISPR is that there are a lot of diseases that are not tied to genetics very well. People have, of course, been searching and searching for genetic links to autoimmune diseases, rheumatoid arthritis, type 1 diabetes. They've been searching for genetic links to type 2 diabetes and susceptibility to it. Alzheimer's. How many searches have been done through the genome looking for Alzheimer's genes? I think we can say there is no Alzheimer's gene. Someone would have found it by now if there were really a reasonable Alzheimer's gene candidate. So what is CRISPR going to do for you for Alzheimer's? We would have to know a heck of a lot more about it before we could go in and start talking about doing some kind of gene editing to improve it. And we just don't know that much. So I think where CRISPR really has the biggest near-term disruptive potential is in small orphan Mendelian type diseases. The interesting thing, of course, is there are a lot of companies targeting those small Mendelian diseases, and they have very big financial plans based around them. So I think everyone who's messing in that area is keeping a very close eye on the gene editing technologies, because that has the tendency, the potential to really shake things up. But it's too early to say when that shakeup is going to occur, where it's going to occur first, and how thoroughly things are going to get shaken once it started.
I have another question. As you might expect, you're getting a lot of questions about the job the job market. This is one about the sort of career ladder and whether it even exists at this point in industrial chemistry. This person is asking whether you would recommend younger chemists go deep or broad in their field of inquiry. Maybe you could tackle that one. We might have touched on that a little bit earlier, but
That kind of gets back to Aristarchus again and the fox and hedgehog stuff. The fox knows many things, but the hedgehog knows one big thing. I think that you're probably better off being a little bit more broad than deep. The thing is, you have to be prepared to go deep or deeper on topics as they come up, targets of opportunity. But I think you should probably keep your default setting more broad, to be looking for ideas, technologies, targets, biology techniques whatever could be affecting drug discovery, I think you need to be looking at that as broadly as possible because ideas can come from anywhere. And a lot of the best ones come from synthesizing two fields. If you're the chemist who knows a lot more about field X than most chemists do, that's an advantage. That is an advantage for you, and it's an advantage for discovering something. I like to quote A.J. Liebling. He was a writer for the New Yorker back in the 40s and 50s. He used to say that his secret was that he was faster than everyone who was better, and he was better than everyone who was faster. Now, he would have made a pretty good blogger if he was serious about that, but that's the sort of sweet spot you need to be into. I think you need to know more as a chemist. You need to know more about some of these biology and analytical technical areas than your average chemist does. But you can't be a deep expert on all of these things. But you can go around and say, okay, here are the ones that I think are actually pretty interesting or pretty neat that I don't see other people messing with around here. Maybe I should learn a little bit more about this so I can decide for myself if this is something good or if I can think up something to do with it. So I guess that's my answer is broad but selectively deep, and be ready to ditch something if it turns out it's not working out, and be ready to hit the literature again if there's some new thing that comes up and say, all right, I have to sit down and learn this stuff. Being a quick study is indispensable. That may be a shame because there are people who are not quick studies, the hedgehog types, who end up really digging into a topic and finding things that you can't find out otherwise unless you dig into it for years. But I don't think that's really happening in drug discovery. I don't think it's been that way historically, and I don't think it's been that way for a long time. So in the same way that the Curious Wave Function blog was mentioning recently, I believe drug discovery is more of a field for foxes than hedgehogs. So keep it in mind for what it's worth.
I'll make this the last sort of chemistry wonky question since we're running out of time, but I wondered if you could weigh in on phenotypic screening. I think there's been an ongoing debate there, and some of the readers are asking questions about, where you fall. What are your thoughts on the future of high throughput screening and what's going to be the most effective?
Well, I like phenotypic screening a lot, but I have to put in a word of caution there. A bad phenotypic screen is the worst of both worlds. That is the abyss. By bad, I mean one that is not necessarily predictive of the disease that you're studying. And that's the hard part. Setting up a good phenotypic screen is really difficult, partly because of the kinds of targets we're studying now, partly it's just intrinsically difficult to find a model on the cellular, small animal, rodent, what have you, to find a biological model like that you are confident enough of to go with. Because when you get down to it, if you're going to be seriously phenotypically screening something, you have to have enough nerve to to screen your compounds in this, to develop your compounds against this, and then when you've got a good-looking compound and you've done a little testing for PK and you've done a little testing for tox and it seems to work in your phenotypic screen, there's nothing left to do. You have to hold your breath, cross your fingers, and go into man. You have to go into the clinic with it, and that's a big leap. That's kind of a broad jump to make. If you're not ready to make that, you need to either steel yourself, screw your courage to the sticking place, as Shakespeare put it, or you need to rethink your commitment to running the phenotypic screen in the first place because you're just going to be wasting your time. So is your phenotypic screen solid enough? Do you have enough confidence in it to be able to make that leap eventually? Because that's what you're headed for. So a good phenotypic screen is a wonderful thing. I don't think we know nearly enough biology we certainly don't know as much as we sometimes think we do. And a good phenotypic screen is the way to discover it, the way to find things out that we didn't even know that we knew. But a bad phenotypic screen is a waste of time. And if it's a really simple, easy thing to set up, the chances of it being a bad phenotypic screen are increased. So look out.
Are there therapeutic areas that you're really looking forward to us making some significant progress on in the coming years? Maybe you could talk a little bit about sort of aspirationally, what problems we might be able to solve in the next few years that you think are really, tough ones. I guess I'm hoping we can end on a aspirational note for drug discovery.
Well, we mentioned immuno-oncology before. A lot of the stuff happening in the oncology and cancer field is very exciting. I'm optimistic about that area. I think that we're getting, we talked about epigenetics. I'm very interested to see what happens in that area. I think there's going to be some big failures, but probably some pretty nice successes too. Maybe not successes that people were expecting or successes in the way that they were looking for, but I'm looking forward to seeing what happens in there. Some of the other areas, Alzheimer's, I don't have as much hope for in the near term. I hope to be wrong about that. I'm hoping that we're getting a little bit better handle on diseases of protein misfolding. That's a good area to be in. Very hard, but there's a lot of stuff out there. Infectious diseases, I think that area, if we get the sort of, terrible virulent plague type stuff that people worry about, then yeah, we're going to go crazy with that. As it is now, it's been very hard. I have a tremendous amount of respect for bacteria because they are so hard to kill. I don't know how many mammalian cells I've killed over the years, but killing bacteria, especially gram-negative bacteria on demand, is very hard unless you're using bleach or something like that. Then it works okay, but bleach is not a very good therapeutic agent. So I would hope for some breakthroughs in anti-infectives, but I don't really know where they're coming from. The good thing is that it's a lot easier to set up meaningful phenotypic screens in bacteria and viral infections. So I'm hoping for good things to come out of that, but it's really kind of a crapshoot. So overall, I guess oncology is the area that I'm most happy about, and the spillover from all the immuno-oncology is just the other immune diseases. So the autoimmune area, and then as you get on from there into the inflammation, so immune, allergy, inflammation stuff, I think we're going to see a lot of spillover from that as we start to learn more about the immune system from all these oncology applications. That area I'm very actually keen on seeing what happens next.
Okay, one last question I think we have time for. This might be too big of a bear for the last question, but I just wondered if you could talk a little bit about the pharmaceutical industry's reputation. Obviously, between drug pricing and adverse events that come up after a drug is approved, there's kind of the industry gets kind of a bum rap, and I wondered if you could talk about what scientists could do to help change that that perception.
I don't know how much we can do. That's one of the reasons I started the blog, is that I found that people that I talked to outside the field had no idea of what drug discovery was like. They had no idea that people even had a job during drug discovery. People still don't. It's too big a mound of ignorance to shovel out so easy. The industry does get a bad rep, some of it deserved. Some of our pricing ideas are not sustainable, and some of the things that we're generating are not providing enough benefit for those prices. However, others are. There are drugs that are expensive that are worth it because they do a lot. That's our challenge, is to come up with things that are worth the prices that we would like to charge for them. Now, as far as the reputation with adverse events, every drug that everyone takes is an investigational drug. Honestly, I would like to see every prescription bottle have a label on it that says, I understand that I am participating in a post-approval trial of an investigational medicine, because nothing is settled. Everything, we're still learning about stuff. Drugs that have been on the market for years and years. So everything is an investigational drug, more or less forever. So adverse events are always going to be with us. We cannot predict these things well enough. The FDA would like for us to be able to, but how big a clinical trial can you run if this is going to be a drug taken by X million people? So some of the bad reputation the industry has deserved, some of it isn't. We as an industry would be out of a lot of our trouble if we delivered some drugs that really dramatically work. People wouldn't complain so much about what they cost if we can deliver real benefit. That's our challenge.
Okay, well, I think that's all we have time for. Thank you so much, Derek, for letting everyone pick your brain. We all appreciate your insightful commentary on the drug industry, and thanks to all of our viewers for tuning in. I think there's still some more talks left on our agenda. You can find the full program on the agenda page at the upper right hand of your screen. And in the meantime, you can stop by the virtual exhibit hall in the lounge to check out our sponsors and exhibitors, or to network with other attendees. And if you want to check back in on Twitter to join the conversation, again, the hashtag is CENSYM15. Thank you so much.